DNA, or deoxyribonucleic acid, contains all of the hereditary or genetic material in humans and most other organisms. So the DNA is what lets people know what genetic material is in a person or is in a certain creature or other organism. Now each cell in an organism has the same DNA. So if someone were to get a piece of your hair and a fingernail and a piece of skin and look at it very closely and look at the DNA in all three would be the same, and it would let whoever was looking at this know that all three of those articles came from the same person. This is the same for other organisms. Each cell in the organism's body is going to have the same DNA pattern so that you'll be able to tell that it came from the same creature. Most DNA is found in the nucleus of a cell. There is some DNA in the mitochondria, and then it is known as mitochondrial DNA, or it would look like this if you saw an abbreviation for it. So if you see the little MT before DNA in the abbreviation, that's because it's mitochondrial DNA and it's found in the mitochondria instead of the nucleus. But most DNA is going to be in the nucleus. Now, that important hereditary information found in DNA is stored as a code made up of 4 chemical bases. We've got adenine, guanine, cytosine, and thymine. And each one of those bases is only going to pair with one other base. They can't all pair up with any of the other 3. So adenine pairs with thymine, and guanine pairs with cytosine to form base pairs. And a way that I remember that is that the ones with the straight lines in their abbreviation letter, the A and T that can be made with straight lines, go together, and the ones that require a curved line go together. And that helps me remember that adenine pairs with thymine, these straight lines to form A and straight lines to form T, and guanine goes with cytosine because of the curved shape that it takes to make each of those abbreviation letters. The sequence of the adenine, thymine, guanine, cytosine pairs determines how the organism builds and maintains itself. So it's important to think about this. Not every organism or every person is going to have the exact same sequence of base pairs. And you might think of the alphabet. We use 26 letters to form all of our words and sentences, but the way that they're arranged, the sequence of those letters is what lets us know what word is being communicated, what sentence is trying to be communicated. And each word has a different meaning, even though it's made up of the same 26 letters or some subset of those letters. So it works the same way. You're going to have some sequence of adenine, thymine, guanine, cytosine, and those base pair sequence is going to let you know, or let a scientist know, what that organism does to maintain itself, what kind it is. Is it functioning properly? Is everything in the right order? What does that order tell us? The same way that seeing CAT tells us that that is the word cat, and it's talking about a mammal with pointy ears and whiskers and a tail that's kept as a pet. Let's look at the next part. Each base pair, so our adenine thymine pair or guanine cytosine pair, is going to attach to a sugar molecule and to a phosphate molecule. And once those attach, once a base pair attaches to the sugar molecule and phosphate molecule, it forms a nucleotide. And these nucleotides form two long strands that spiral into a double helix. That is the shape that DNA takes on. And I did my best to give y'all an example of a double helix. It kind of spirals around. If you were to curl a ribbon around my pen, it would form that kind of double helix shape we're talking about. And you should think of a twisted ladder with the base pairs as rungs. So if we were looking at this picture here, this would be our double helix shape that's spiraling around, and each one of these little rungs on the ladder would be a base pair. So we would have adenine and thymine together, and they would be paired up. And then here you would have guanine and cytosine paired up. And then you would have adenine and thymine, guanine and cytosine, all the way down the ladder, where you've got your sugar molecules and phosphate molecules that bound to your base pairs, making up the sides of the ladder here. And you just have this long DNA chain, and then the sequence that you see is going to let you know how that organism builds and maintains itself, because not every creature's DNA is going to be the same. Everyone has a very unique DNA pattern. DNA can replicate or make copies of itself by splitting the ladder in half. So if we're looking at this and we just decided to go through and cut this ladder in half, we would be separating those base pairs all the way down. So then you have AG, and any other base pairs you've got. So we know we've got adenine, guanine, and because we know what's on one side of the ladder, each strand or half of that ladder serves as a pattern for duplicating bases. Since we know we've got adenine here, we know the thing that has to pair with it is thymine. If we've got guanine, cytosine has to pair with it. Since in DNA, adenine and thymine always pair together and guanine and cytosine pair together, you will know, or the cell will know, okay, I've got this DNA, I've only got half of it, but it's a pattern. If I have adenine, I need to add thymine to the other side. If I've got cytosine, I need to add guanine to the other side. So the cell will know, based on that half, that strand that it gets from DNA, how to make a complete DNA molecule and be able to form that double helix and complete the DNA strand. And each new cell needs an exact copy of DNA from the old cell. Because remember, each cell in an organism has the exact same DNA. So if the DNA is replicating itself, it needs to be giving an exact copy to a new cell so that this can continue, and the organism is going to continue to have the same DNA in it. So you will see a lot about DNA in biology, and it's important to remember that DNA is the hereditary or genetic material. It's usually found in the nucleus. And remember which bases pair with which. Your straight lines in adenine, A, pair with your straight lines in thymine, T, and your curve G for guanine goes with your curve C for cytosine. DNA is the hereditary or genetic material that is usually found in the nucleus, sometimes in the mitochondria. The most important thing to remember is that it's where all the genetic material is going to be found. It's a big identifier for a cell or for an organism. Genes. A gene is a molecular unit of heredity of a living organism. A gene is the basic instruction, a sequence of nucleic acids. So a gene is going to be the instruction, it's going to be the sequence of nucleic acids that tells the cell to code for a certain protein or for a certain RNA chain. It isn't just one little thing, it's a sequence that gives an instruction to the cell. While an allele codes for one variant of that gene. So if you've heard someone say, oh, she has really good hair genes, or she's got the gene for blue eyes, that's not exactly true, because everyone has a gene for eye color or hair color, and for every other trait that you have. But what determines what color your hair is or what color your eyes are is going to be the alleles for that gene. So there are dominant alleles and recessive alleles. And if you have even one dominant allele, then you're going to have the dominant gene. If you have two recessive alleles, then you will have that recessive gene. For instance, if you have two parents that have brown eyes, you could still end up with blue eyes because blue is a recessive gene. So both of your parents had one dominant and one recessive brown and blue eye color allele. The brown would have been prevalent in both of them. But since they both had that recessive blue allele present, you could still end up with blue eyes because of the recessive alleles combining. And I know that all sounds a little confusing, but it's just to tell you that each gene doesn't determine something all on its own. It's the alleles that are the variants of that gene that determine what the outcome of each gene sequence is. Genes do specify all proteins and functional chains. So they specify how these are going to be created, the functional RNA chains. Genes hold the information needed to build and maintain an organism's cells and pass genetic traits to offspring. So where I was talking about two parents that could have brown eyes and still pass on the blue eye color, it's because of the alleles that they carry, whether dominant or recessive, whether you can see it as a physical trait or not. Your parents carried genetic traits and they passed those on to you. And some of your physical traits may not all be the same as one or both of your parents, but the genetic traits that you got did come from the alleles that they had combined and mixed together. So you took some of your mother's alleles and some of your father's alleles, and when those combined, you ended up with different alleles and different genetic traits of your own, but they both came from the genetic traits your parents had. Some examples are eye color, number of limbs, and blood type. So some genes you can see right away manifested, such as eye color or number of limbs. But blood type is something that you would have to do a little more scientific research to discover. You can also search for certain markers in your body for diseases such as Alzheimer's or other hereditary diseases. Those can be tested for, and your DNA will show, your genes will show if you have a likelihood of getting that disease. And so that's definitely not something you can see just by looking at someone. It's something that you would have to do more research into. But it's possible by looking at the genes in your body. Now, for a gene to create or code for its protein, it must go through transcription. And transcription is where single-stranded messenger RNA or mRNA is created that is complementary to the DNA from which it was transcribed. And we'll look at that over here in just a minute. But basically, you're starting with DNA and you're transcribing it into the messenger RNA. And then the messenger RNA is going to bring the message elsewhere in the cell. So next comes translation. That messenger RNA is used as a template for synthesizing a new protein. Transfer RNA, or tRNA, brings the amino acids and anticodon necessary for protein synthesis. And something to remember is that genes are read 3 nucleotides at a time in units called codons. So you look at the 3 nucleotides that are coding for this one protein, and they're going to be transcribed onto messenger RNA, which is going to travel elsewhere in the cell, and transfer RNA is going to bring the amino acids and the anticodon that match up with that set of 3, with that codon, and let the protein be created. So let's look at that over here. Let's pretend this gene sequence came from a strand of DNA. And remember, your bases are adenine, guanine, cytosine, and thymine. So we are going to say it's going to change from DNA to the messenger RNA. So we're transcribing here, and each base must be transcribed. So here we've got our transcription. So the guanine is going to transcribe to cytosine, cytosine to guanine, thymine to adenine, cytosine to guanine. There we go. Thymine to adenine and thymine to adenine. So the messenger RNA is going to carry this sequence. So you can see how it isn't exactly what was on the DNA, it's what base would have paired with the DNA. So it's kind of like a code you've got to sit here and figure out. You've got to decode what the messenger RNA says. So the messenger RNA has the pairs that would have gone with the DNA, and so in comes your transfer RNA, which is going to have its amino acids and the anticodon that's necessary for each of these. So each set of 3 is a codon, and it gets transferred over. And so now the transfer RNA is going to come in, and it's going to bring what we need. So it's going to say, okay, we had cytosine, so now we need the guanine. I guess we should really point this way. And then it's going to bring cytosine back to pair with the guanine, thymine back to pair with adenine, cytosine back to pair with guanine, thymine to pair with adenine, and thymine to pair with adenine. So now in a different part of the cell, you've got these codons, and they're going to code for a certain protein. And this is what happens in the cell. So this part would be your translation. So you've got strands of DNA. Part of the cell says, oh, I need a certain protein. Well, this codon codes for one of the proteins you need, and this codon codes for one of the other proteins you need. So the DNA is going to send it. The messenger RNA is going to hold the transcription, which is where it's changed to what the pairs would be. What pairs with each base is going to be the transcription that goes on the messenger RNA. And then the messenger RNA travels off into the cell to where the protein is needed. The transfer RNA is going to bring the bases plus amino acids. And then you're going to get these bases plus the new amino acids are going to give you your proteins that these codons were coding for. So you've got your transcription where it's coded onto the messenger RNA, and then you've got translation where it's decoded and the transfer RNA brings in what you need plus amino acids and your proteins are created. So that's kind of a complicated process, but it's what genes do. That's their job. Their job is to send messages throughout the cell so that new proteins can be created. And they are going to carry your hereditary information, and they have the basic instructions necessary to tell your body how to carry out the hereditary processes and keep producing the genes that you keep producing the cells and the proteins that you need and maintaining your cells. So just keep in mind, genes are units of hereditary information, and they carry messages throughout the cell and instruct the cell on how to create the proteins that your body needs. Kingdom Animalia. Animals have several characteristics that set them apart from other living things. Animals are eukaryotic and multicellular. So where other kingdoms can have creatures that are comprised of just one cell, animals are going to have more than one cell. They are also heterotrophic. This means they cannot create their own food and must rely on other sources of nourishment. So they're going to eat other plants or animals. They're not going to be able to create their own food with sunlight like plants can. Animals do not have cell walls, but they may have an exoskeleton, which is like a hard skeleton they secrete onto the outside of their body, or a shell. Now, they don't have to have that. Obviously, we don't have a hard exoskeleton or a shell, but some animals like your crawfish or lobsters would have an exoskeleton, or things like snails have a shell. All animals are motile. This means they can move, even if only during certain stages of life. So some animals may only move a little bit or very slowly, but all animals are motile, even if it's just for a short period of time or during one stage of their life. Nearly all animals undergo some form of sexual reproduction, but some animals are also capable of asexual reproduction. And this is possibly, and a lot of times, through parthenogenesis, which is when a fertilized egg is produced without any mating, budding, or fragmentation. So it is possible, but most of the time the animals are going to undergo sexual reproduction to produce offspring. Animalia is also known as metazoa, as far as a kingdom name goes, and it is divided into the subkingdoms, Parazoa, which consist of sponges and trichoplax. Sponges, I'm sure you can think of, but trichoplax kind of looks like a puddle you spilled on the floor. It's very flat, but it is still an animal. It does have differentiated cells. It even has some eye cells on it. So Parazoa, such as the sponges and trichoplax, have differentiated cells but no distinct tissue or tissues or body symmetry. So they're kind of oddly shaped. Where most animals you would be able to see some symmetry, there is no symmetry as far as Parazoa are concerned. Now, all other animals are put into the subkingdom Eumetazoa. And these have 2 or 3 distinct layers of cells with differentiated tissues and either radial or bilateral symmetry. And what that means is that you can divide them a certain way. If they have radial symmetry, you would be able to divide them any way going in a circle, and the halves would match up. So a jellyfish would be an example of that. If you divided the jellyfish here, if you could divide it across here, if you could divide it across here, going any way in a circle around the top of the jellyfish, down across the top and down through all the tentacles, if you cut it, it would be more of a circular shape, and anywhere you cut it, it would be the same on either side. So it's symmetrical. And it would have radial symmetry because there isn't just one symmetrical line you can cut along. There's not just one line of symmetry, there are several. The same goes with a starfish. You could cut it along a line going this way, 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 or a line going this way. If you go all along that circle, it can have symmetry. It can have all these different lines of symmetry, and what's on the other side of the line of symmetry is going to match what's on the first side of the line of symmetry. Bilateral symmetry, you can only divide the body in half one way. So people have bilateral symmetry. You can divide straight down from the top of your head to between your 2 feet, and the 2 halves should match up pretty well, at least as far as body type and tissues are concerned. So people have bilateral symmetry. You can divide straight down the middle and the halves should match up pretty well. Butterflies or moths are another example that should match up. Cats, dogs, a lot of animals display bilateral body symmetry, but there are some that have radial symmetry. But these are all Eumetazoa. Remember, the Parazoa is not going to have any kind of body symmetry because it doesn't have distinct tissues, and it is not going to have a distinct body shape every time. It's going to be different. So whenever you are thinking about kingdom Animalia, remember these certain characteristics that set them apart. They're eukaryotic and multicellular, made of more than one cell. They are heterotrophic. They cannot make their own food and must receive nourishment from other sources. They do not have cell walls, but may have an exoskeleton or a shell. And all animals are motile. They are able to move, at least at some stage in their life. Meiosis. Sexual reproduction in eukaryotes involves a form of cell division known as meiosis. It has several different stages that are carried out to let one diploid germ cell result in 4 haploid gametes. And diploid means that it's going to have multiple copies of the genetic information, where haploid means it's going to have one copy. So, in this first part, this would be interphase. And it's kind of before meiosis starts, but it has to do with it because this diploid germ cell contains genetic information from both a maternal and paternal parents. So you've got female and male genetic material in this one diploid germ cell, and it's getting ready to divide to carry out sexual reproduction. So we have replication going on in here because each chromosome is going to double itself so that there's extra genetic material to carry out this process. In the next phase, we have prophase. And the replicated genetic material is going to line up in homologous pairs, which means that each one is the same genetic material. It's an exact copy. And they're going to cross over a little bit at these points here. And where those cross over, they kind of swap a little bit of genetic material. So each one of these pairs is going to take a little bit of the other pair. And that is called crossing over or recombination. So you're just kind of jumbling up the maternal and paternal bits before the division starts to take place. Then we move on to metaphase. And in metaphase, your centrosomes are starting to pull the homologous pairs apart. And then we move on to anaphase. And you can see they actually are getting pulled further apart. They're not crossing over anymore, and they're starting to move to opposite sides of the cell. And these are all prophase 1, metaphase 1, anaphase 1, because this is kind of a two-step process. Then we keep going, and we've got telophase 1. In telophase 1, there's the cleavage furrow that's preparing for the cells to separate and become two distinct separate cells. There is a nucleus being reformed. The microtubules that have been connecting the chromosomes and pulling them apart have receded, and the genetic material is encased in a nucleus again. And then this last part is cytokinesis 1, where the cytoplasm is split, and there is a definite split made between those two cells. So now there are two diploid daughter cells. So we had our diploid germ cell. The DNA, the genetic material was all replicated, so there was twice as much. We went through this cell division, so now each one of these daughter cells is a diploid daughter cell, and it has twice as much genetic material as it needs to be a haploid gamete. So we're going to go through what is known as meiosis II, the second step in this process. So we have prophase 2. And in this phase, you'll see we've got the two daughter cells, and now they're kind of repeating the same process that happened up top. So there's no replication because that was an interphase, but they are going to start having the centrosomes move down to either side and start moving out to opposite poles of the cell and start pulling the chromosomes that way. In the next phase, we've got metaphase 2. You can see that the chromosomes have lined up along the equatorial plate in both of these daughter cells. The microtubules are connecting them to the centrosomes, and they're going to start pulling apart. So in our next phase, anaphase 2, you can see that actually happen. The chromosome pairs have gotten pulled apart, and now these individual chromatids are on separate sides of the cell, and they're still going to be kind of being reeled in by the microtubules at this point. And we move on to telophase 2, where we've got a nucleus forming around our new pairs of genetic material that have both the mother and father genetic material in each nucleus. They're all split up now, and we have the cleavage furrow again. The cells are getting ready to divide permanently. Your centrosomes have separated to the opposite side still, and the microtubules have retracted. And then we go to cytokinesis 2, which is where the cytoplasm divided between these two new cells and these two new cells, and now we've got 4 haploid gametes, which means that they each just have one set of genetic information, and they're going to need a partner to be able to reproduce. So these gametes are going to be ready to be fertilized, and when they are fertilized, a diploid zygote will be formed. So these gametes are ready to be fertilized, and if they are, then they can go on to create a new eukaryotic organism. So meiosis does have a 2-part process, and it goes through the same phases as mitosis, but they're a little bit different because instead of just getting an exact copy, you're mixing genetic material from 2 different organisms, a maternal and paternal parent, and at the end you're going to have the 4 haploid gametes that each has genetic information from both parents. Mitosis. Eukaryotic cells reproduce asexually by dividing into 2 genetically identical daughter cells. Now they do this by the process of mitosis. So let's look. This first phase is called interphase, and it's not really considered a part of mitosis, but it's usually discussed with mitosis because it happens at the beginning right before mitosis happens. It's the build-up to mitosis. It's a period of cell growth and DNA replication. So the cell gets a little bigger, the nucleus gets a little bigger, the DNA replicates, so there's more of it for the mitosis that's about to happen. Chromosomes are in the form of uncondensed chromatin, and the nucleolus may still be present. So this would be our uncondensed chromatin. This darker dot here would be your nucleolus that you could still see, and these little pieces here would be your centrosomes. So whenever you see them along the rest of mitosis, remember these are the centrosomes. Okay, first stage of mitosis is going to be prophase. In prophase, the chromatin condenses into discrete chromosomes. So remember, it had gotten we had uncondensed chromatin that the DNA had been turned into. In prophase, it condenses into discrete chromosomes. So instead of a bunch of DNA squiggled up, you've got discrete chromosomes that kind of look like little squiggly X's. The nuclear membrane breaks down So where we had a complete nuclear membrane here, it starts to break down a little bit, and sister chromatids are joined by centromeres. So what they mean by that is this would be your chromosome, and these sister chromosomes or chromatids are joined by these centromeres. So this would be your centromere. And then each of these would be sister chromatids that form the chromosome. The centrosomes are going to move to opposite poles of the cell. So remember, we just kind of had them in the cell here. Now they've moved to opposite sides of the cell, so they're opposite each other and they're kind of equally spaced in the cell. The chromosomes have paired up to form sister chromatids connected by a centromere. And now we're ready to move on to our next phase, which is metaphase. In metaphase, the chromosomes align at the equatorial plate. So where they're kind of just all over the place, they've lined up at the equatorial plate, which means that it's equally distanced from both of those centrosomes that have spaced themselves out at either pole. The microtubules attach sister chromosomes to the centrosomes. So you've got your centrosomes, and these are your microtubules that are attaching the chromosomes to the centrosomes. They've lined them all up along this equatorial plate. And now we're ready to move on to the next stage of mitosis, anaphase. In anaphase, the centromeres divide. So remember, your centromere was that little center here that was kind of holding the two chromatids together. So in anaphase, they divide. The microtubules shorten, so where they're longer, they shorten up, moving paired chromosomes to opposite sides of the cell. So we had these paired chromosomes, and now the microtubules are shortening. They're moving those individual chromosomes over closer to the centrosomes now. And then you're ready for the last phase of mitosis, which is telophase. In telophase, the cytoplasm divides along a cleavage furrow. And so you can see how it starts to kind of dip in. It cleaves in half, and so there's that cleavage furrow, and that's going to show you where your cytoplasm is going to eventually split completely off and form two new cells. The chromosomes condense to chromatin. Remember, that's what we had up here. We had all the squigglies. They're going to just start condensing to chromatin, not a discrete chromosome anymore. And the nucleolus reappears, and the nuclear membrane is going to be reforming around a clear nucleus. So you see the nuclear membrane is forming around where your condensing or your uncondensing chromosomes are, and your nucleolus has reappeared. So once these cells split off, then this is going to be a nucleus again that looks much like this, with the uncondensed chromatin and the nucleolus in the center. Two daughter cells, as we discussed at the beginning, two identical daughter cells will be formed once cytokinesis is complete. And cytokinesis is that process of the cell being cleaved in half to form two new identical daughter cells. So this is mitosis. It is how a eukaryotic cell can reproduce asexually. It just divides into two identical daughter cells. It goes through interphase, prophase, metaphase, anaphase, and telophase. So it starts off with this uncondensed DNA, this uncondensed chromatin genetic material. The chromosomes form little pairs and the centrosomes come to either side. The centrosomes attach to the centromeres with microtubules, and those chromosomes line up along the equatorial plate. Then they split apart, starting to look like two new cells now, and each part of the chromosome pair goes toward a centrosome so that they are split equally across the cell. And then they split apart, forming a new nucleus with a nucleolus, and cytokinesis is going to be complete once this cell finishes cleaving in half. So just remember that eukaryotic cells can reproduce asexually by mitosis. Photosynthesis. Photosynthesis is a process used by plants and other autotrophic organisms to convert light energy, usually from the sun, into chemical energy that can be used by the organism. Photosynthesis usually gives off oxygen as a waste product as well. Oxygen is not something that the plant or other autotrophic organism will need in most cases, and so it will give off oxygen as a waste product. Now, autotrophic means that these organisms make their own food. They are capable of creating their own food. They can harness the light energy and create their own source of nourishment. Heterotrophs cannot do that and must rely on autotrophs or other heterotrophs for their nourishment. Most plants, most algae, and cyanobacteria perform photosynthesis and are called photoautotrophs. So, autotrophs are organisms that can create their own food. Photoautotrophs are organisms that create their own food from light, since photo translates to light, looking at the Latin root for that word. These are organisms that can create their own nourishment from light. Photosynthesis maintains atmospheric oxygen levels and supplies most of the energy necessary for life on Earth. So, as these autotrophic organisms take in carbon dioxide, they let out oxygen and help maintain the atmospheric oxygen levels, because people and other organisms exhale carbon dioxide, and the plants or other autotrophs take that carbon dioxide in and turn it back into oxygen, which we breathe. So if we didn't have photosynthesis, we'd be running low on oxygen. And so photosynthesis helps maintain our oxygen levels. It also supplies most of the energy necessary for life on Earth. Even though you don't feel like you're eating light or getting your energy just from light, if you eat a vegetable, then you're eating something that it has only been able to grow because it used light energy to provide nourishment for itself. So you are now eating that plant, which got its energy and was able to grow from light. If you were to eat a cow, if you had beef, if you had a hamburger, then you would be eating another heterotroph. Cows can't make their own food, but they ate grass and other vegetables and other foods that were grown, and they were grown by having light energy harnessed inside those plants. And so the cow got the energy from the plant, which got the energy from the light, and then you were getting energy from the cow. So it all comes back to the light energy. So while there are other sources of energy, the light energy is a primary source for everything going on Earth. Photosynthesis always begins when energy from light is absorbed by proteins that contain green chlorophyll pigments. So The autotrophs that perform photosynthesis don't always look green, but somewhere in their body, they're going to have these chlorophyll pigments that are able to take in and absorb light to start the process of photosynthesis. And photosynthesis occurs in two stages. You have your light reactions or light-dependent reactions, so these depend on light to happen. And it's when organisms capture and store the light energy. The light energy is converted to chemical energy in the form of NADPH and ATP. So in this light reaction or light-dependent reaction stage, the light energy is actually captured, turned into a chemical form of energy that can be stored or immediately used if necessary. In the carbon dioxide fixing reactions or light-independent reactions, these are ones that can happen even whenever light isn't present, as long as there was some energy harnessed in that plant already. So these don't have to happen while there is light present supplying energy. They can happen later. And this is when the formed NADPH and ATP, so it's already stored and formed in that autotroph, those two energy molecules drive the reduction of carbon dioxide to more useful organic compounds, such as glucose. And they are also referred to as the Calvin or Calvin-Benson cycle. So when the carbon dioxide is reduced, that process can be called the Calvin or Calvin-Benson cycle as well. So even though this is a light-independent reaction, it does still depend on molecules that were created by converting light energy into a stored chemical energy. So even though we call them light independent, they really couldn't occur if you didn't have the light transferring, the light energy that was able to be transferred to these energy molecules that are stored chemically. The main thing to remember about photosynthesis is that it is the process by which autotrophs can make their own energy using light. Rocks versus minerals. Let's look at a side-by-side comparison of rocks versus minerals so you can see the difference between the two. Rocks are aggregates of one or more minerals, and they may also contain mineraloids and organic remains. Now, mineraloids are minerals lacking a crystalline structure, so they don't have a definite crystalline structure if they are mineraloids. And organic remains are going to be anything that came from a living thing. So if an animal died, then its body would eventually decay and be organic matter. Any kind of excrement from an animal would be organic remains. And so all of that also can form up rock. So a rock is an aggregate or a combination of one or more minerals, and it could contain mineraloids, and it could contain organic remains. So there's no real set combination of what a rock is going to contain. And it has no definite structure or shape. So a rock doesn't have to have a certain structure. It doesn't have to look like a certain shape whenever you find it. Rocks are more random. You're not going to find them looking the same way every time. Now, minerals are naturally occurring inorganic solids, so nothing organic about them, nothing that came from a living creature or a person. And they have a definite chemical composition. So if you were to look at it, every atom of that mineral should have the same chemical composition and an orderly internal crystalline structure. Now, some minerals may have the same chemical composition but have a different crystalline structure, and the structure determines how it's going to look or what the mineral is going to be called sometimes whenever it has the same chemical composition. And let's see, so that's a mineral. A mineral at its basic point is a naturally occurring inorganic solid, so made naturally from the earth, and it doesn't have anything that used to be alive. It has a definite chemical composition and a definite internal crystalline structure. So it's got to form the same way every time to be a specific mineral. And a polymorph is two minerals with the same chemical composition but a different crystalline structure. So this is what I was talking about, where sometimes, even though two minerals have the same chemical composition, they won't look the same because their crystalline structure is different. And at that basic atomic level, if the crystalline structure is different, what you're able to see with the naked eye is going to appear different. And even if it looks like two different minerals, it isn't going to be the exact same. It'll be a polymorph of the other because it doesn't have the same crystalline structure, even though it has the same chemical composition. So rocks are more random. They don't have to have a definite structure. They don't have to have a definite chemical composition or makeup. It can be made of any combination of minerals, mineraloids, and organic matter. Minerals are going to have a definite chemical composition and an orderly internal crystal structure. So they are more specific. They have to have a definite composition and structure to them, where rocks do not have to have that. Now, rock types are igneous, sedimentary, and metamorphic. So these are the three rock types that a rock could be classified as, and rocks are classified into one of these three types based on their formation and the minerals they contain. So how were they formed and what are they formed of? When you know those two things, you can classify your rock as igneous, sedimentary, or metamorphic. Minerals are classified by their chemical composition. So you look and see what chemicals it's made up of, what exact atoms make up that mineral at a chemical level, at the tiniest level, and that's how they're going to be classified. Rocks are classified by how they are formed. Are they formed from stress? Are they formed from heat? Are they formed from organic matter? Those kind of things are going to affect how a rock is classified, but that doesn't matter for a mineral. And lastly, the study of rocks is known as petrology. And the study of minerals is known as mineralogy. And side note, geology is the study of Earth as it pertains to the composition, structure, and origin of its rocks. So, rocks are classified based on their formations and what minerals are in the rock. Minerals are classified by their chemical composition. At that basic root level, what are they made up of? Petrology is the study of rocks. Mineralogy is the study of minerals. Rocks are more random. They don't have a definite structure. They're made up of one or many different minerals plus mineraloids and organic matter. Minerals are more specific. They have to have a definite chemical composition and a definite crystalline structure.
In a chemical equation, it is necessary to have the same number of atoms for each element on each side of the equation. So right here, we have a salt. And so we have three numbers here. We have a big 2, smaller 2, and a smaller 4. And so this big 2 right here is called a coefficient. And these smaller numbers are referred to as subscripts. And so they serve different functions. So a coefficient tells how many molecules, and a subscript tells how many atoms. And so when you're trying to balance a chemical equation to make sure you have the same number of each type of atom on the left and right side of the equation, you cannot change the subscripts because the subscripts control how many atoms are in that molecule, and so that's the particular makeup of that molecule. For example, if we change this 2 right here to a 1 on sodium, it would totally change the molecule we're dealing with. But if we change this 2 all the way up to an 8, it wouldn't change the makeup of this molecule because this right here is just the moles of this molecule. It's how much of this molecule we have. And so it would just mean that we have 4 times as much of this molecule if that number changes. So right here, we have a great example of how to balance a chemical equation because we have diatomic hydrogen combining with diatomic oxygen to form water, H2O. Now, the reactants are always on the left side of the equation, and the products are on the right side of the equation. So these things over here are reacting together to form this product or products. And so the reactants are always on the left, and so we need to count how many of each atom we have on this side. So we have a subscript of 2 here, so we have 2 hydrogens, and we have 2 oxygens. Now over here on the right, we have a subscript of 2, so we have 2 hydrogens, and we have 1 oxygen. Now this one doesn't have a subscript at all, but it's just an understood 1. So we could write a 1 right there. But just to make things easier, instead of writing a 1, it's left out many times. And so in order to balance this, we can't change the subscripts, because the easiest thing to do would just be to change that 1 to a 2. But we can't do that because we'd alter the chemical makeup. And so we have to alter the coefficients. So what we can do here is put a coefficient of 2 in front of water. So now we have 4 hydrogens over here, and we have 2 oxygens. So now the oxygens are equal, but the hydrogens are not, because now we have 4 hydrogens over here. So we can put a coefficient right here, and so we have 2 moles of this hydrogen molecule, and so now we have 4 hydrogens total. So we just multiply that coefficient by the subscript, and so that is equal as well. So that's how you balance a chemical equation. I want to take a look at 4 of the basic properties of gases. And I want to talk about the properties of gases in the context of a balloon so that it gives us something to visualize when we're talking about these properties. So think of a balloon, and as you blow air into this balloon, you're adding molecules to the air. As more of this air gets added to the balloon, the volume is going to increase, which makes sense. Everyone knows when you blow air into a balloon, the balloon expands. And if you let air out of the balloon, then the balloon gets smaller. So volume is proportional to the amount of gas. Now, for this to be true, the temperature and the pressure has to be constant. And you may be wondering, why is that? Well, you'll see later that temperature affects the volume and the pressure affects the volume. So if the temperature and pressure are changing, then they're also affecting the volume in addition to the amount of gas, and we can no longer really see what's really affecting the volume. So volume is no longer proportional to the amount of gas. But if temperature and pressure are constant through this entire time, we can pretty much take them out of the equation because they're not affecting anything anymore because they're not changing. So when temperature and pressure are constant, we can then say volume is proportional to the amount of gas. Now, the second property of gas is also very interesting. If you think about a balloon that has a fixed amount of air in it, in other words, the end's tied off. You can't add air, you can't remove air. And then you heat this balloon, it will expand. The volume will increase. As you heat it, the balloon will get bigger. Now, if you cool the balloon, then the volume will decrease. The balloon will get smaller. So we can say the volume changes directly with temperature. As temperature increases, volume increases. And as temperature decreases, volume decreases. Now, for this to be true, the pressure of the balloon has to remain constant throughout the entire process. Now, if you squeeze the balloon, the volume will decrease because gases are compressible. So we can say the volume changes inversely with the pressure. And this makes sense. Okay, so say there's lots of pressure. You're pushing really hard on this balloon. The volume is going to decrease. So at that point, the pressure is increasing because you're pushing harder, but the volume's decreasing. And if the pressure decreases and you're not pushing as hard, then the volume will increase. So that's why we say volume changes inversely with the pressure. And for this to be true, the temperature has to remain constant. Now, the fourth and final property of a gas. If you fix the volume of the balloon and add more air, then the pressure inside will increase. So we can say pressure changes directly with the amount of gas. And this makes sense because you think about air molecules inside that balloon. They're moving all over the place. Well, if volume, or if the amount of gas is large, then the molecules are going to hit the sides very often. Okay, so you think about the volume being the same throughout this whole time. The balloon is always going to be the same size. But if you add more air to it, then there's more air molecules that are going to hit the side of the balloon more often. So that increases pressure. But if there's less air inside that balloon, then the air molecules are going to hit the sides of the balloon less often, which decreases the pressure. So that's why we say pressure changes directly with the amount of gas. Now, for this to be true, like I said, the volume has to be constant and the temperature also has to be constant. So those are the four basic properties of gases. Light is the portion of the electromagnetic spectrum that is visible because of its ability to stimulate the retina. Light is absorbed and emitted by electrons, atoms, and molecules that move from one energy level to another. So an atom might release energy to move to a lower energy level, but absorb energy to move to a higher energy level. Now, visible light is between ultraviolet and infrared light on the spectrum. So it's anywhere from 380 nanometers all the way up to 760 nanometers. So 380 would correspond to the color violet, and 760 would correspond to the color red. This is because the human brain interprets or perceives visible light as color. For example, when the entire wavelength reaches the retina, the brain perceives the color white. Now, when no part of the wavelength reaches the retina, the brain perceives the color black. Atoms and molecules can gain or lose energy only in particular discrete amounts. Therefore, they can absorb and emit light only at wavelengths that correspond to these amounts. So, using a process known as spectroscopy, these characteristic wavelengths can be used to identify substances. We can classify cells into two categories, prokaryotic and eukaryotic. So prokaryotic cells are smaller than eukaryotic cells. In addition to being smaller, they lack membrane-bound nucleuses. So they don't have a membrane-bound nucleus. Now, they contain single-stranded circular DNA molecules that free float in, you can probably guess, they free float in cytoplasm, because cytoplasm is kind of the fluid in a cell that all the other organelles in the cell float around in. So the DNA molecules free float in cytoplasm. So some different organelles that are in prokaryotic cells are ribosomes, centrioles, often one or more flagella, and cytoskeleton elements. Then we have eukaryotic cells, and so these are going to be obviously larger than prokaryotic cells. And these contain linear, double-stranded DNA combined with histone and packaged as chromosomes. And then the chromosomes are found inside a nucleus bounded by two membranes. So notice here, the eukaryotic cells do have a membrane-bound nucleus. Now, eukaryotic cells contain a variety of other membrane-bound organelles, such as mitochondria, chloroplast, endoplasmic reticula, Golgi bodies, lysosomes, and vacuoles. Now, eukaryotic ribosomes are larger and more complex than those found in prokaryotes, which is what is another way of saying prokaryotic cells. Now, you may be wondering, where do you find prokaryotic cells and where do you find eukaryotic cells? Well, prokaryotic cells are usually in single-celled organisms, such as bacteria, whereas eukaryotic cells are what you find in more complex organisms. It's what you see the result of on a daily basis, because plants and animals are made up of eukaryotic cells. So that's a look at prokaryotic and eukaryotic cells and the differences between them. The solar system consists of the sun and the 8 major planets that orbit around it. So first we have the sun, and then I'm going to order the 8 major planets in order of how close they are to the sun. So we start out with Mercury, then Venus, then the third from the sun is Earth. Then we have Mars. And then in between Mars and the next planet, we have an asteroid belt. Then after that asteroid belt is Jupiter, then Saturn, Uranus, then Neptune. Now you may be thinking Pluto should come after Neptune. Well, Pluto is no longer considered to be a planet. Now it's considered to be, or is classified as one of the 5 known trans-Neptunian objects regarded as minor planets. So then after Neptune, we have the Kuiper Belt. And so this Kuiper Belt extends from about 30 astronomical units from Neptune to about 50 astronomical units. So an astronomical unit is the distance from the sun to the earth, and so that's 150 million kilometers. So the Kuiper Belt extends from Neptune at 30 astronomical units out to about 50 astronomical units. So that's a total of 1 light year, or 9.5 trillion kilometers from the sun. And so this contains countless numbers of icy bodies of water, methane, and ammonia, some with rocky cores. Now beyond the Kuiper Belt lies the hypothesized spherical Oort cloud, thought to be the source of long-period comets. So that's the Oort cloud. And so there's 8 major planets, so I'm going to put a check mark next to those. And again, these are in order from the distance they orbit the sun. So notice here that if you remember any kind of certain phrase to remember the order of the planets, notice that there's 2 M's. And so Mercury is the closest to the sun, and then Mars is after Earth. But all the other planets start with different letters. So the order is Mercury, Venus, Earth, Mars, Jupiter, Saturn, Uranus, and Neptune.
Today we're going to briefly go over one simple machine, the wheel and the axle. The wheel and the axle, categorized as a simple machine, and we want to talk about mechanical advantage of the wheel and axle. Essentially, with a wheel and an axle, most people tend to think of automobile tire, because that's usually what we talk about, is a wheel and an axle. 2 wheels attached by an axle. But what we want to think about to help illustrate the point of the mechanical advantage of the wheel and axle is something more along the lines of a steering wheel. Essentially, the steering is done by the central column. But if you had to grip that central column and turn it with your hand this way, it would be almost impossible. So by attaching a wheel to that central axle and moving the radius of that circle out, it takes much less force to turn the central axle and therefore to turn the car. So what you have then is a mixture of one type of force, the wheel, attached to the axle, making it easier then to do what you would ordinarily have to exert a great deal of force if you grabbed the main axle itself and tried to turn it by itself. So I've got a picture here. This is the central axle here. This is the outer wheel of the steering wheel, if you will. If we had to grab the central column and turn it, it would be almost impossible for us to get enough force. But you move the wheel out, attach the axle, and you apply force out here, and it becomes much easier to turn here. So force here translates here to greater force. So the mechanical advantage increases with the radius. So I've got an example up here. If we've got a radius of 12 versus a radius of 10, with our axiom over here, The mechanical advantage increases with the radius. Bigger radius, 12, means greater mechanical advantage. What we mean by that is if the same amount of force was applied to the outer wheel at 10 inches and the same amount of force applied to the wheel at 12 inches, you would get a much greater advantage at the 12-inch radius than at the 10, assuming the same amount of force is applied. It becomes, it translates here to the central axle much greater. So the mechanical advantage is greater the larger the wheel is. And in the same way, if you've got a larger wheel, it takes less force to turn it. So if you think about it, sometimes in little sporty cars, they have sort of smaller steering wheels, but when you get to something as big as a bus or an 18-wheeler, they usually have very large steering wheels. And this is just an application of this simple machine, the wheel and the axle, the mechanical advantage increasing as the radius also increases. Well, this has just been a basic overview then of the simple machine, wheel and axle. If you'd like to learn more about this or related matters, underneath this video, you'll find a link. If you'll click on that link, it'll take you to that website that has that information. And while you're on that website, you'll also find a link to an ebook that's ready for immediate download.
An experiment is a set of actions and observations performed to confirm or not confirm a hypothesis or to research a causal relationship between phenomena. Now, only one condition in an experiment is changed at a time. So the conditions that affect the outcome we call variables, and then the factors in experiments that do not change are called constants. So we're dealing with variables and constants. So again, variables are the factors that do change, and constants are the variables that do not change. Now, experiments have two parts. We have the control group and the experimental group. If we just had an experimental group, we would not be able to determine anything from the observations of an experiment. That's why we need a control group, which is the group under normal conditions. So nothing unusual is done to it. And then we have the experimental group, which is the test group in which variables are changed. So the experimental group is the most important, but we have to have a control group to understand what's going on in the experimental group. So in other words, if you're changing variables here, you don't know what effect they're having because you don't have a normal group. So you start out with a group under normal conditions, and then you go to the experimental group where you only change one variable at a time. And so by changing that one variable, you get to compare the differences between the experimental group and the control group. So we're looking at cause and effect here, because you have one variable that's going to cause other things to happen. So we start out with the independent variable. Which is a manipulated variable in an experiment or study whose presence or degree determines the change in the dependent variable. And so then that leads us obviously to the dependent variable, which is the observed variable in an experiment or study whose changes are determined by the presence or degree of one or more independent variables. So we have an independent variable and a dependent variable. One's manipulated and the other is observed. So the independent variable is the variable you're changing. Remember, I said you can only change one variable. So you're changing the independent variable, and we're looking at cause and effect. So the independent variable causes an effect with the dependent variable. So that's what you're looking at here, the cause and effect between the independent and dependent variables.